Hello everyone, my name is Grace Lehnhoff and this is my presentation on the anti-cancer properties of cannabidiol via effects on estrogen metabolism. So first we'll be starting talking about ovarian cancer as a whole. Ovarian cancer is a very deadly disease that begins in the ovaries. With that being said, a woman has a chance of 1 in 75 of a diagnosis of ovarian cancer. As a woman increases in age, the diagnosis of ovarian cancer rises. The American Cancer Society states and predicts that in 2021, about 21,410 women living in the United States will receive a new diagnosis of ovarian cancer. This can also be seen with the prediction that about 13,770 of those same women will die from ovarian cancer. Now on to our lab's purpose. While being in Dr. Hales' lab, I have been able to research the white leghorn hen, which has a very similar reproductive system to that of a human's reproductive system. And this can be seen in many different diseases such as ovarian cancer. We use the hen model to research preventative measures for ovarian cancer, and we look at this pathologically and histologically. As for my project, our main goal is to detect the expression of genes that onset and prevent ovarian cancer. When looking at cannabis, CBD has been highly researched over the past many years, and it is around 1 of 60 cannabinoids in the cannabis plant. It can be used as a high anti-inflammatory and anti-anxiolytic agent. And as we research the properties of CBD, we come to question, will cannabidiol ingestion target ovarian cancer inflammation? This could potentially lead CBD to be anti-carcinogenic with research supporting it. As I get to talking more about CB receptors, we will see that CBD is a CB receptor antagonist. This will lead us into the purpose of this study, which is to quantify the anti-carcinogenic effects of CBD on estrogen metabolism by assessing ovarian morphology and the biochemistry of the liver. Now, let me tell you about my project that I've been working on this summer. I've been working with a gene called CYP1A1, which hydroxylates estradiol to form 2-hydroxyestradiol. This eventually gets methylated by the enzyme catechol-O-methyltransferase, or COMT, and this has been shown to decrease the probability of the formation of ovarian cancer. My hypothesis for this project is if each group of hens has a fixed increasing dose of CBD, then CYP1A1 will be increased. This will cause a dose-dependent increase in 2-methoxyestradiol along with a dose-dependent decrease in inflammation. Now on to the second part of my project. There is another aspect of CBD that will be researched, and it is the CB1 and CB2 receptors. These are a part of the endocannabinoid system, which is a complex cell signaling system which responds to these cannabinoids. The CB1 receptors are found in the central nervous system, and the CB2 receptors are found in the peripheral nervous system. My hypothesis for this project is if each group of hens has a fixed increasing dose of CBD, then expression of CB1 and CB2 receptors will increase. So this brings up the question of how will this be measured? Starting from the animal study, we pilled the hens with CBD every day for 1 month and took part in wing vein bleeds at the end of every week to collect the blood plasma in these hens. The tissue harvest then took place, and we performed a necropsy to harvest the tissue samples used in this study, which are ovaries and livers. RNA isolation was then performed to obtain RNA samples from the tissue by homogenization. This led to cDNA synthesis, which we used a thermocycler to obtain cDNA from the RNA that was isolated. Then we used the cDNA for qPCR, which is used to show the expression of certain genes in samples, and the gene that I will be looking at is CYP1A1, or the CB1 and CB2 receptors. So on to CYP1A1 mRNA expression measured by qPCR in the ovary. When looking at the data on this slide, the melt peak on the right tells us that the reference primer works well in our ovary samples. This reference primer was RPS2, and you can tell it works because of the strict parabolic curves within the same limits. This reference gene, RPS2, can be used in calculations to indicate how much of the CYP1A1 primer is present in the sample. Looking at the graph on the left now, this tells us the CYP1A1 mRNA expression via the laying hen ovary. As you can see, the error bars overlapping indicate CYP1A1 expression is non-significant in the ovary. So on to CB1 and CB2 mRNA expression measured by qPCR in the ovary. While using the same ovary samples and the same RPS2 reference primer, I quantified the CB1 and CB2 expression in the laying hen ovary. Both of these graphs indicate that the data is non-significant and doesn't really show much of a difference between the designated groups. So now on to CYP1A1 mRNA expression measured by qPCR in the liver. This shows a different story. Another reference gene was found that worked best for the liver samples, which again is RPS2. This reference primer was used again in the calculation for the CYP1A1 in the liver. This graph on the left shows almost exactly what we want to see, which is really good. This graph supports my hypothesis, which is also really exciting, of a dose-dependent increase of CYP1A1 in the liver. Although this data is still non-significant because the error bars overlap, it's really exciting to see the data and it being on the right track of my hypothesis. So in summary, the conclusion was that CYP1A1 gene and the CB1 and CB2 receptors were measured in the ovary, and CYP1A1 was also measured in the liver. From the trials that were done, the data was non-significant, but CYP1A1 has a dose-dependent increase in the liver. For my future directions, CB1 and CB2 will be quantified in the liver, and immunohistochemistry will be used to examine CYP1A1 in the liver and ovary. The liver, ovary, and blood plasma will also be measured for 2-methoxyestradiol. Prostaglandins will be quantified by ELISA to see if inflammation is decreasing. And these prostaglandins are inflammatory mediators. So looking at these inflammatory mediators will determine if inflammation is decreasing or increasing. So this ELISA will tell us if that is happening. And the techniques that I have learned are protein assays, H&E staining, histological identification, Western immunoblotting, quantitative polymerase chain reaction, and RNA and DNA isolation. So I'd like to thank SIU Simmons Cancer Institute staff and generous donors, SIU School of Medicine, Dr. Buck Hales and Dr. Karen Hales, Dr. Chris Weston, and a plethora of undergraduate researchers. Thank you all.